Hello everyone. Welcome to today's session on role of adipose tissue and liver in lipid metabolism. Myself, the facilitator, Dr. Mausmi Shankar Adala, working as an assistant professor in Department of Biochemistry, St. Joseph's College for Women, Autonomous, Visakhapatnam. Today's topic are included under the subheadings: the roles of adipose tissue and liver in the metabolism of lipid. There are various fields in which we explain about their role. Some of them are adipokine secretion, insulation and protection, fatty acid release, lipid storage, and thermogenesis. We'll be always covering the facilitating energy availability by releasing fatty acids, the primary role of adipose tissue in storing energy, regulation of the metabolism and inflammation through hormones-like substances, provides thermal insulation and protects internal organs, and finally generates heat, especially in the brown adipose tissue. Introduction. Lipid metabolism is a crucial physiological process involving the synthesis, storage, and degradation of lipids, which serves as vital energy sources and structural components of cells. Two major organs, the adipose tissue and the liver, play central roles in maintaining lipid homeostasis. Adipose tissue primarily acts as the body's main energy reservoir. It stores excess energy in the form of triglycerides during the times of energy surplus and releases free fatty acids during periods of energy demand through a process called lipolysis. Beyond energy storage, adipose tissue also functions as an active endocrine organ secreting various hormones and cytokines, namely adipokines, that influence appetite, insulin sensitivity, and systemic metabolism. The liver, on the other hand, orchestrates the complex processing and the distribution of lipids. It is responsible for the synthesis of fatty acids, that is lipogenesis, the assembly and the secretion of lipoproteins such as very low density lipoproteins, VLDL, and the oxidation of fatty acids to produce energy, especially during fasting. The liver also plays a critical role in cholesterol metabolism, balancing its synthesis, uptake, and excretion. Role of adipose tissue in lipid metabolism. Firstly, the storage of lipids. Adipose tissue primarily stores energy in the form of triglycerides, that is the fat molecules. When there is excess energy intake, more calories than the body needs, adipose tissue absorbs the fatty acids and glucose from the blood and converts them into triglycerides for long-term storage. Release of fatty acids. During fasting, exercise, or energy deficiency, adipose tissue breaks down stored triglycerides into free fatty acids and glycerol. These products are released into bloodstream and transported to muscles, liver, and other tissues for energy production through beta oxidation. Secretion of adipokines. Adipose tissue is not just a passive fat store, it is an active endocrine organ. It secretes hormones and signaling molecules called adipokines like leptin, adiponectin, and resistin that regulate appetite, insulin sensitivity, inflammation, and overall lipid metabolism. Insulation and protection. Besides metabolism, adipose tissue provides thermal insulation and mechanical protection from internal organs. Regulation of lipid homeostasis. Adipose tissue helps maintain lipid balance in the blood. Dysfunctional adipose tissue, such as in obesity, can lead to elevated free fatty acids in the blood, contributing to insulin resistance, fatty liver, and atherosclerosis. Energy storage. Stores excess energy as triglycerides during periods of energy surplus, that is after meals. Each adipocyte, that is a fat cell, can expand many times its original size to store more fat. Coming to lipogenesis, that is a fat formation. In the fed state, adipose tissue takes up glucose and fatty acids. Glucose is converted into glycerol, and together with fatty acids, it forms triglycerides through a process called as lipogenesis. Lipolysis, which is nothing but the fat breakdown. During fasting, exercise or stress, hormones like epinephrine and cortisol stimulate lipolysis. Triglycerides are broken down into free fatty acids and glycerol, which enter the bloodstream to supply energy. Fatty acid uptake. Adipose tissue captures fatty acids from circulating lipoproteins such as chylomicrons and VLDL through the action of an enzyme called lipoprotein lipase, abbreviated as LPL. Remodeling and expansion. Adipose tissue can remodel itself by increasing the size, that is a hypertrophy, or number, that is hyperplasia, of fat cells depending on energy needs. Thermogenesis in brown adipose tissue. Brown adipose tissue specializes in burning fats to produce heat, especially important in newborn and during cold exposure. It contains many mitochondria and expresses uncoupling protein 1, UCP1, to generate heat instead of ATP. Regulation by hormones. Insulin promotes lipid storage by increasing glucose uptake and lipogenesis while inhibiting lipolysis. Catecholamines, that is similar to adrenaline, which stimulate the lipolysis during stress or exercise. Production of adipokines, which is a signaling molecules. Leptin regulates appetite and energy balance by signaling the brain about fat stores. Adiponectin, it enhances the insulin sensitivity and promotes fatty acid oxidation. Resistin and others influence insulin resistance and inflammation. Buffer against lipotoxicity. Adipose tissue helps buffer and traps excess fatty acids, preventing harmful lipid accumulation in non-adipose tissues like liver, muscle and pancreas. Finally, once again, thermogenesis, which is present in the brown adipose tissue. It is specializing in burning fats to produce heat, especially important in the newborns, and also expressed as uncoupling protein 1, abbreviated as UCP1, to generate heat instead of ATP. With this, the first half of the topic, that is the role of adipose tissue in lipid metabolism, was covered. Moving on to the next topic, role of liver in lipid metabolism. Firstly, the synthesis of lipoproteins. The liver produces very low density lipoproteins, that is VLDL, that transport triglycerides and cholesterol from the liver to other tissues. It also contributes to the production of high density lipoproteins, HDL, which removes excess cholesterol from the tissues and transport it back to the liver, that is a reverse cholesterol transport. Fatty acid oxidation. When the energy is needed, the liver breaks down fatty acids into acetyl-CoA by beta oxidation. This acetyl-CoA enters the Krebs cycle for energy or be used to produce ketone bodies. Lipogenesis, that is a fatty acid synthesis in the fed state, especially after a carbohydrate-rich meal. Excess glucose is converted into fatty acids in the liver. These newly synthesized fatty acids are assembled into triglycerides and packed into VLDL particles for transport. Cholesterol synthesis and regulation. The liver synthesizes cholesterol, a vital component of cell membrane and precursor of steroid hormones and bile acids. It regulates blood cholesterol levels by balancing synthesis, uptake from the blood and excretion into bile. Ketogenesis, which is nothing but the ketone body formation. During the prolonged fasting, starvation or low carbohydrate intake, the liver converts fatty acids into ketone bodies, namely acetoacetate, beta-hydroxybarbiturate and acetone, which serves as alternative fuels for the brain and muscles. Regulation of blood lipid levels. The liver maintains plasma lipid balance by controlling the production and clearance of lipoproteins and free fatty acids. Detoxification of lipid-derived molecules. The liver helps in detoxifying lipid peroxidation products and removing excess lipid-soluble substances like fat-soluble vitamins when in excess. Bile production. The liver produces bile salts from cholesterol, essential for the digestion and absorption of dietary fats in the intestine. Storage of the lipids. Although adipose tissue is the main fat storage site, The liver can temporarily store fats, especially triglycerides. Excessive accumulation leads to fatty liver disease, that is hepatic steatosis. Storage of the fat-soluble vitamins. The liver stores fat-soluble vitamins like A, D, E, K and regulates their release as needed. Very low density lipoproteins recycling and LDL formation. VLDL secreted by the liver loses triglycerides and is converted into IDL and then LDL, nothing but low density lipoprotein, the major carrier of cholesterol in blood. Handling of oxidized lipids. It detoxifies oxidized lipids to protect against oxidative stress and inflammation. Response to insulin and glucagon, that is the hormones. Insulin stimulates lipogenesis and inhibits fat breakdown. Glucagon promotes fatty acid oxidation and ketogenesis during fasting. Support in acute phase response. And stored temporarily. Uptake and processing of chylomicron remnants. After dietary fats are absorbed and packed into chylomicrons, the remnants after the fat delivery to tissues are taken up by the liver for recycling or disposal. Regulation of lipoprotein metabolism produces enzymes and apolipoproteins like ApoB100, ApoE, ApoA1 that regulate the metabolism and function of lipoproteins in the blood. Fatty acid desaturation and elongation. The liver modifies fatty acids by adding double bonds, that is desaturation, or increasing chain length, that is elongation, creating diverse types of fatty acids needed for cell membranes and signaling molecules. Synthesis of phospholipids. These are essential for building biological membranes. The liver produces phospholipids like phosphatidylcholine and phosphatidylserine. Generation of lipid-based signaling molecules. Produced important molecules like prostaglandins and leukotrienes from fatty acids which are involved in the inflammation and immune response. Regulation of energy homeostasis senses the body nutritional state and adjusts lipid metabolism accordingly, promoting fat synthesis in fed state and fat oxidation, ketone production during fasting. Concluding today's session, lipids and adipose tissue play a central role in lipid metabolism, acting as both key sources and storage sites of energy. Lipids, primarily in the form of triglycerides, serve as dense energy reservoirs that fuel various physiological processes when metabolized. Adipose tissue not only stores these lipids but also functions as an active endocrine organ, releasing hormones and signaling molecules that regulate appetite, insulin sensitivity, and overall metabolic homeostasis. The dynamic interplay between lipid mobilization, storage, and utilization underscores the critical importance of these components in maintaining energy balance and metabolic health. Disruptions in lipid metabolism or adipose tissue function can contribute to metabolic disorders such as obesity, diabetes, and cardiovascular disease, highlighting their significance in both health and diseases. Thank you one and all for your support and cooperation.